Well, I'd like to share my thanks to Willie and to Sam for the invitation and the organization of this really fascinating symposium. A lot has been Mind the Gap. I changed my title slightly, so I guess this goes forward. Yeah. The gap that we're going to be minding is that gap between guard cells, which of course regulates gas exchange. Now, historically, an enormous amount of information has been accumulated over the past three or more decades with the onset of patch clamping techniques, which were, in fact, one of the founders of the patch clamp on guard cells is going to be the next speaker. And therefore, we have a huge body of information about ions and channels and pumps and ATPases and so on, and a lot of information in addition about regulation by ABA and so on, and environmental factors. But the cytoskeleton has been somewhat neglected, and that's going to be, I'll talk a little bit about something else first, which is the role of 1433 protein in stomatal function, a little bit on ubiquitination of stomatal function, but then the main part of the talk will be related on the role of microtubules. Now, just a retroapatite, with red light alone, that guard cell pair is closed. You can't see the gap. I had to fake it because only the fluorescence of the tubulin is what you see there. It's GFP tubulin. And when they're open, you can see the changes that occur. And it's obvious that there are things happening. If you now want to do a time course of stomata that have been, guard cells that have been treated with ABA, you'll notice that when they're open, now this is better resolution, you get, which is the pointer? Yeah, here. You've got a huge number of these structures which are composed of one or more microtubules. Whether it's true bundling or whether they're just a loose association isn't clear at this point. But notice that as in this case, they were put in the dark and then imaged every 10 minutes, and these are 3D reconstructions. You notice two things. First, that the number of very bright ones goes down, but also that there's a loss in total fluorescence. Now, since the tubulin monomers are actually perfectly stable and continue to fluoresce, this can only mean that there is degradation of the tubulin occurring during stomatal closure, and that there will be, therefore, have to be resynthesis of that tubulin on opening. Okay, let's just talk quickly about this 1433. I think you all know that a particular serine will get phosphorylated, and then that binds the 1433, and that can now be involved in activating an enzyme. Lots and lots of examples of this. And the question, of course, is what does this scheme have to do with guard cells? And a postdoc, Dongchung Seng, was doing yeast-2 hybrid and picked up a very nice interaction with 1433 lambda isoform. A very strong association. He did pull-downs, which I won't show, but they all were positive. And then he asked, well, which domain might it be? Here you find that if you use the phototropins, whoops, go back. Phototropins have, as you know, two domains here which bind flavin, and these are the photoreceptor regions, these chromophore domains, and a downstream kinase. And when you activate particularly this chromophore domain, the protein opens up and activates the kinase, and downstream various signal transduction pathways are activated. And it looks as if the kinase now is the region that is somehow interacting with the 1433 protein. If we then ask, well, where in that kinase is it? There are these binding motifs for 1433, and it turns out that at 747, there is what appears to be this classic motif for binding. And here he had enormously good luck, because there are lots and lots and lots of serines, and we know from other studies that fully 15 different serines and threonines get phosphorylated when you turn the light on this molecule and saturate it. So anyhow, serine 747, he simply decided what happens, let's take a look, is whether that's needed or not. And now if you put the kinase domain in with the serine knocked out and substituted by an alanine, you lose the association. So it looks as if this is at least one place where the 1433 binds. And it was known from Shimazaki's lab that 1433s are required both for the activation of an ATPase mediated by phototropin and, in fact, interaction with the phototropin itself. So it comes as no surprise that one now can localize at least one 1433 binding site. So now, of course, does it have any physiological impact? And the answer is yes, it does. It reduces stomatal opening dramatically. This is now done in a FOAT1 background, so we're dealing with just FOAT2 functional. Otherwise, we'd never see this phenotype. But notice that when 747 is mutated to alanine, there's very much reduced stomatal response to various different intensities of blue light. And then we can complement, put it back in with the wild type and restore the response. So it does look as if it's important for that. Well, this is just to show that the levels of expression were, in fact, approximately the same. And now we can put together a little scheme like this, that the phototropin becomes phosphorylated with blue light. It's dephosphorylated, at least FOAT2 is, by a PP2A. That we didn't talk about, but we published that a couple of years ago. But then there's this 1433, which is required for activation of the blue light response for stomatal opening, but not for phototropism or chloroplast movement. It's highly specific. A, it's specific to FOAT2, and B, it's specific to that one response of Arabidopsis, namely stomatal opening, and not phototropism. Okay. Now, what else is going on with the phototropin? With the help of Xiang Wang and, again, one of his postdocs in Tongsheng, we did some mass spec on this, in this case on FOAT1, and picked up what appeared to be a ubiquitination site at the position lysine 527, as you'll see up here, right there. And this was, now one might ask the question, well, is that ubiquitination necessary for anything, and where is it in the molecule? There it is. And now if we look at the nice solution structure from Kevin Gardner's laboratory, Shannon Harper did this nice work, you'll notice that this is the domain itself. It's folded over, it's got 5 beta sheets and some alpha helices. And then downstream is this so-called J alpha helix, which is amphipathic. That is, one side is hydrophobic and is somehow maintained tightly against the beta sheets. And when you irradiate this, there is a tug inside and some photochemistry I won't talk about, which then allows this to be flipped open to the C-terminal and activate the kinase. Now, there's a tryptophan right here, and when you shine blue light on this thing and then watch the tryptophan, its fluorescence moves out to a longer wavelength because it's being moved into a more polar environment. And then when the thing closes up again, which it does, that fluorescence moves back to a shorter wavelength. And that's exactly where that lysine is, so that it looks as if there may be, we have reason to believe it's a monoubiquitination, which is playing some sort of a signaling role. Well, let's take a look at that. Whoops. Is it? What is it doing? Again, what happens if we mutate it away? Do we get any kind of a phenotype? And the answer is yes. If we mutate it away, we get a very dramatic reduction in the stomatal opening response. This is changing 527 to arginine, both 26 and 27 to arginine. The other line is just coming along, so that we'll have 26 alone. But it looks as if ubiquitination somehow, and it looks from other studies as if it's monoubiquitination, is playing some sort of a signaling role. Okay, well, it also is playing a role in phototropism. Here again are those same 2 transformants mutated, and there's considerably less phototropic curvature in response to the same intensities of blue light compared to the wild type. And then that's just another one. Well, now let's go back. Those are some of the biochemical things that we've been looking at, but now let's take a look at those microtubules. And the first thing we noticed in this now is work that was started by Bill Eisinger. He was at Santa Clara University, but he spent many long days, weeks, hours in my lab because he didn't have the opportunity to do that at Santa Clara, and he just recently retired. But in the meantime, he showed that, for example, the number of these microtubule structures that you see when the guard cells are apart and the stomata are open is closely correlated with stomatal aperture. So these are just plants that were taken out of the greenhouse, and he measured the stomatal aperture, and then he counted the number of these structures he could see. If they're closed, you notice that there's a big reduction in the number, and a reduction, as I mentioned, in total fluorescence. So then you can show that if you start with them all wide open and follow them in time as they close, there's a decline in, there's a label over here that just disappeared, but this would be the number of the aperture, and you see that the aperture declines in parallel with the number of these structures that you can resolve. And then if you take a look at the loss in fluorescence, fluorescence in this particular experiment went down about 50%. There was a slight increase in guard cell cytosolic fluorescence, but notice that this is a 10-fold amplified to put it on this scale. So there's a real genuine loss, a major loss in fluorescence. In the pavement cells, nothing much happens. And again, as you induce them to close, and this, I believe it was ABA, you get a decrease in stomatal aperture. And then you can take them from the light into the dark, and then put them back in the light, and again, the correlation holds. So, of course, I think I won't bother with that one. One of the things now that you might ask is, well, why is the reduction in these microtubules, and how is it mechanically, how is it happening? As you can imagine that it might be much more rapid degradation at the minus ends. It might be a real slowdown in synthesis at the positive ends, or some combination. And so let's take a look first at the number. And for this, I want to go back to, oops. Yeah, hope this works. There's another label that we had, fortunately. Is that what I want? I somehow don't think so. I knew that was going to happen.
I don't know why it didn't do this one.
Oh, yes. Yeah. Okay, this is end-binding protein 1. This is a different EB1, I think, one we heard about a little bit earlier today. But you'll notice that, I have to make it play again. You can actually count these guys. And you see, this is now three-dimensional, so you're looking at, you're looking at these comets. And they're all moving at roughly the same speed. But now you can look at them in open or closed guard cells. You can count them. And now can I possibly get back without screwing up? Ah, come on, here. Yeah. You know, if you look at the number of them now between the guard cells and the pavement cells, starting with guard cells, stomata that are open and inducing them to close with ABA, there's no difference in the number. So that doesn't account for it. It isn't that you have less growing ends. Well, maybe they're going slower. That's another possibility, if they're moving more slowly. But if we check the rate of movement, and you can do that too, of course, from these images, you'll notice that there's no difference in the rate of movement when the guard cells are open or when they're closed. There's no difference in the number of these things. So therefore, you have to conclude that what we're seeing is the result of massive degradation at the minus ends, and resulting in ultimate loss in total tubulin. So, less tubulin overall, no change in the number of growing ends, no change in the average rate of growth, and therefore it has to involve degradation somehow. Now, the next question, of course, is, does this have anything to do with function? Are they playing a role? And if you modify them somehow, can you change opening and closing behavior? So this is now a study using a couple of drugs. One of them is Orizolin, which destabilizes microtubules, and the other one is Taxol, which stabilizes them. And now we have both Fischer-Faba and Arabidopsis, and just looking at stomatal aperture. And if they're initially in the dark, of course, the aperture is very small. Put them in the light, up they go. And then, but if you put them in the light in the presence of Orizolin, they don't open. So and you can see what the cell looks like. Whereas if you put them in the light in the presence of Taxol, up they go, and you get these beautiful bundles forming, or, well, associations. I don't quite dare call them bundles yet. Now we can do the opposite experiment. Start in the light, go into the dark, and they both close, as you would expect. If you close them, try to close them in the dark in the presence of Orizolin, of course they close, not surprisingly. But now if you put them in Taxol, they're stuck open. So this would suggest then that the microtubules are in fact playing some sort of a functional role in the stomatal mechanism, without defining precisely what that is. We wanted to make certain that our various labeled transformants with GFP or YFP or whatever weren't affecting normal stomatal function. So this is simply gas exchange measurements of all of the various transformants, showing that when the light's off, the guard cells in the leaf close, lights on, they open up again, and water loss goes up, and then light off again. No really significant, so it's a good control. So then we could get a little bit more quantitative, and this first efforts were rather crude, simply scanning along this line and plotting brightness. And you see there are big peaks and little peaks, and so that valley would correspond to this dark place, and so on. And then if you take a look in time, there are 6 guard cells represented here. And when they're open, this is what you see from the 6 scans in terms of fluorescence intensity. And notice that after 10 minutes is less, 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 and finally all those bright bands are gone when they're closed. On the other hand, if you look at the pavement cells, you see nothing of the sort. There's still plenty of bright bands. And so it looks as if something really different between pavement cells and guard cells. Then Bill got more sophisticated, and I guess I should say Dave Earhart got a better microscope, thank goodness. And now one could take an image like this and draw a line from one end of the guard cell to the other down the median, and then rotate this thing so that you looked at the z-axis. That is, you looked at it end on. So these would represent the ends, the cross sections of those bright bands. And then you could get a scan that had much, much higher resolution, as you can see here. And then if you take these and put them, simply rank them by height, you'll notice that there's sort of a stepwise pattern here. We always saw that stepwise pattern, and I think, say that here's some more of them. And you'll notice, particularly on these, there are really relatively clean steps up by height. If in the darkness, it looks like this. If you have it in the light, it looks like this. If you have it dark back in the light, it looks like this. But one might interpret that as these are singles, these are doubles, triples, quadruples. So that's our present working hypothesis, and that is that we're seeing increased synthesis as they open of tubulin, and that the tubulin is then forming microtubes that progressively aggregate to form these bright structures that we see. Well, now, why does the fluorescence decline on stomatal closing? As I pointed out at the start, tubulin doesn't lose its fluorescence as a monomer. It's still there. So there obviously is an overall loss in tubulin. And of course, proteolysis makes good sense. So now this is some work by Rajneesh Khanna, who just joined the lab recently. And Rajneesh then treated these cells with MG132, which is an inhibitor of the proteasomal pathway, as I think you all know. And so it should inhibit this loss. And if it inhibits the loss, that means that the degradation of the multiple microtubule structures might be delayed, and this might slow the closing response. Also, it was known that COP1 mutation, and that's an F-box protein, was known to prevent stomatal closing, and it might also prevent the loss that we've seen. So let's take a look at that. And here is our 3-repeat experiment. Oh, go away. Yeah, here are 3 repeat experiments in which we're looking at relative fluorescence, either DMSO treated or treated with MG132 for an hour before ABA was added. And you'll notice that in the DMSO only, you get a good drop in fluorescence, much like what we've seen in all 3 these different experiments. Whereas if you have applied the MG132, you get varying degrees of inhibition of the MG132 of the fluorescence loss. And then if you continue on to look at the effect of MG132 on stomatal aperture, here is DMSO, and again, these 3 experiments. And here is 0 time before adding ABA. ABA induces closure, and therefore the stomatal aperture goes down. But if you do exactly the same experiment, you notice that there was no closing of the stomata in any of these 3 experiments. They're all just the same in the, at 0 time as they are at 40 minutes. And then finally, oops, too many buttons to press. If we look at the COP1 mutant, stomatal aperture we know stays open, and in the COP1 mutant, the stomatal aperture stays open, and the fluorescence, in fact, fluorescence, as far as we can tell, and we haven't measured, it doesn't change because nothing seems to change. These guard cells are just stuck very wide open, and they have very, very well developed microtubule structures. So there's clearly a lot more to do. One of the questions we have to address now is, what is the role of these microtubules? It's obvious that as the guard cells expand, either the membrane is all infolded in one case and it's opening out, as accommodating the increase in surface area, or you have to add new material. And also, it is now going to be possible, I think, we managed to convince Julian that it would be lots of fun to look at some of the things he's going to be talking about, but under some of these conditions where we've been seeing these cytoskeletal changes, so that we can maybe put 2 very different stories and approaches together into some kind of model that will, in fact, bring the microtubules into the guard cell picture more clearly. Much of what we've seen so far is observational. There are, of course, many things that we can do, I think, to put these 2 disciplines together and maybe elucidate a bit further what's going on. But we can't possibly do it without our next speaker. So thank you for your attention.
Thank you, Winslow. So before the next speaker, we should have some questions. Willie, let's start.
Winslow, it's simply amazing how you keep pushing us in new directions. Well, it's just amazing. So, but we are talking about cortical microtubules, right? Mm-hmm. And we are teaching our students that it's actually the influx of potassium. That's the potassium exchange, what is involved in the closing and opening and closing of the stomates. And of course, I mean, if you have a potassium influx to drive open or the potassium exchange to drive and close the aperture, then microtubules have to be in place to accommodate that change in the cell structure, right? Yeah. So if you're now saying that during closing the microtubules are degraded and then have to be resynthesized during opening, then how do you accommodate that with the potassium exchange?
One of the hand-waving models that you're putting in channels for delivery of material to the expanding membrane, and what those things might be, whether they're potassium, whether they're channels or whether they're channels or pumps or whatever. is there a trafficking now that's involved that's important to accommodate the expansion of the membrane with the opening response. But, I mean, I think we're in a position that we may be able to test those hypotheses, or hypotheses like that now, but it's sheer speculation at this point, because it is a very interesting question. What the devil are these guys doing?
Any other questions, Winslow? I have one quick question. Do you have any evidence that there's a transcriptional response when you need to resynthesize? So if you need to reopen, do you see any change in tubulin gene expression?
Well, I'm hoping that Rogenus is doing that right now.
Okay.
The question is whether there are any changes in gene expression. In other words, is there some kind of transcriptional role in the opening response or closing? And it is known that there are two MIB transcription factors, one of which is required for optimal opening and another one for optimal closing. So that would suggest that something else is going on. And again, we now are constructing the plants that will have the tubulin and also the MIB mutations. We have them both. So we'll be able to see there, and of course we can look at transcriptional and translational inhibitors as well.
So you've only shown us tubulin. Have you also looked at actin?
Actin. No, but there is a fairly large literature, particularly Young-Suk Lee in Korea, did a lot of studies on actin changes. And so there are obviously big actin changes going on. Now, she didn't do functional studies, so one doesn't know whether those are simply the consequence of opening to bringing about some reorganization, or whether they too play a functional role. So that's another area that badly needs re-exploration.
Okay, thanks again.